Okay, thank you for the introduction. Actually, it was supposed to be the presentation by the researchers in the field, but unfortunately, he has to cancel at the last moment. I just jumped in, and today I'd like to share some updates from my research group on the topic of quantitative phase imaging and artificial intelligence. And this is a dream and hope of my group and many other groups where the quantitative phase imaging technique has been developed. So this is the way that we study the cell morphologies and for the study of the cell biology and also for the diagnosis of various diseases. It has been done for more than 100 years. And I believe there much room to be improved in other to see the better the images for the study of the understanding of the pathophysiology of the cell. And as you may know, there are the variety of microscopic imaging technique from the bright field microscope, fluorescent, the light sheet, the super resolution, phase contrast, and they all have some pros and cons. Bright field microscope is the oldest technique. It's good for imaging cells and tissues upon the stain process. But if you directly look at unlabeled cells or bacteria, they look almost transparent because of the most of the proteins are very transparent in visible wavelengths. And that's why people introduce the staining the agents such as H&E or the pap smear in serology and histologies. I mean, recently people have used the fluorescent proteins in order to specifically label the proteins or molecules inside of the cell by using transfection technique. Although they can provide very high molecular specificity, but you have to worry about cytotoxicity and the genetic modification to the cell. On the other hand, the phase contrast microscope is very nice, label-free technique, has been used for many decades, but it does not provide molecular specificity and it does not provide the three-dimensional information or quantitative information. What I mean by that, you only have the outline or shape of the cell membrane or the bigger subcellular organelles such as nucleus membrane, but you don't have quantitative numbers in terms of the imaging, such as cellular concentrations and volume and etc. And nowadays, the microscopic field is being aided by the development of the artificial intelligence imaging analysis technique for computer-aided diagnosis or the correlations of the multimodal information. And also, by the help of the big data approaches, now people are trying to do better than before by combining this microscopic technique to the artificial intelligence. And my group and many other groups in the world, the concept of quantitative phase imaging has been developed and studied for a few decades. And the idea is to use refractive index as the intrinsic imaging parameter. In physics, the refractive index determines the speed of light as the light is passing through the sample, as Peter has introduced in his talk. In biological meaning of refractive index is protein concentration. And if you use three-dimensional quantitative phase imaging technique, you can reconstruct three-dimensional distribution of this refractive index information. It can provide the morphological information about individual cells as well as subcellular organelles. And same time, you can retrieve quantitative information about protein concentration. So now you have both morphological information as well as the biophysical information. And since it does not require any preparation, you don't have to do fixation, transfection, or staining, which means cells can be monitored and imaged with the healthy stage. And since the speed of limit of this QPI is only limited by the camera speed and modulator speed, which is very fast. So we can study the various phenomena. Sorry for this Korean text, but in short, there are the variety of the QPI technique, like the tychography that Sina will explain in the afternoon session, and the diffraction phase microscope, and Peter has explained, And there are like 2D or 3D techniques, but recently our group is focusing on 3-dimensional QPI technique. It is also called optical diffraction tomography, and we also named like the hollow tomography system, in short HT, for the quick the name and easy name. This is the optical version of X-ray CT, computed tomography system. So in CT, you measure multiple 2D X-ray images as a function of illumination angles, and then the computer reconstruct 3-dimensional distribution of X-ray absorptivity, from which medical doctors can do make decisions by knowing that which part is bone and the tissue and etc. At HT, we use laser beam, coherent laser beam, such as from the Heine lasers or the diode lasers. And using holography or interferometric technique, we measure the hologram images as a function of illumination, various illumination angles. And from which we can reconstruct 3-dimensional distribution of refractive index. So those are the images that we have generated, such as the primary cultured neuron cell. And this is the movies of the NIH 3T3 cell line. And we can also look at the stem cell, neuron cell cultures. And the spatial temporal resolution is very high. So we can go down to 110 nanometer in XY spatial resolution. So now you can see the alterations in morphologies of the individual platelet upon the activation. And the temporal resolution is like tens of 100 milliseconds. So you can do the fast dynamics. By combining both the HT and fluorescent microscope, you can do correlative study. So by measuring the refractive index distribution, you have information about the morphology of the cell. And by simultaneously measuring the fluorescent information, you can have access to the molecular information. So we thought it is crucial to commercialize the system so that it can be used by the clinician and cell biologists, not by the physicists like myself, in order to further utilize this technique. So about a couple of years ago, we have commercialized the HT system. Then it has been used in various research groups. So we have found very interesting application of QPI. The first one is the phase separation or biomolecular condensation. As the Evin has the nicely presented in its talk, since the biomolecular condensate has different concentration, and that can be easily imaged and monitored and also further quantified by using the refractive index distribution. So the Professor Lee in Seoul Regional University also has used this HT machine in order to study the effect of the molecules to the formations of the biomolecular condensate. And lipid is also the interesting topic of the study, because refractive index of lipid is distinctly higher than that of the cytoplasm. So by simply measuring refractive index distribution, you can differentiate what part of the cell is lipid and the other one is cytoplasm. But if you rely on conventional imaging technique, you have to do fixation of the cell, which means the cell is not live anymore. And you have to introduce the staining of using Nile, lead, O, and other chemicals, which is sometimes toxic and not very efficient in terms of the staining efficiency. So you only able to take the still shot of the dead cell, but if you use the refractive index measurement, you can do time-lapse imaging. Since the formation of lipids is very crucial to various diseases such as atherosclerosis and the metabolic disease of the liver cell, and a group in KBSI Korea, and they have used the HT machine in order to quantify the effect of the drugs targeting the formation of the lipids in foam cell in live stage. So this is the interesting topic of the study in QPI. Since we have very high resolution and the advantage of using the label-free refractive index parameters, it can also be used for the study of the microbiologists. And if you use conventional bright field microscope for the study of the bacteria, they all look almost alike because of their similar size and shape. you're only able to tell like the presence of the bacterium, but it is very sometimes difficult to tell which one is established, what dead cell, or the live bacteria. But if you measure three-dimensional tomogram of the refractive index, you can nicely see the presence of the bacterium, and you can also see the growth of the bacteria over time. And furthermore, we have developed the AI-based algorithm to classify the bacterial species based on the three-dimensional tomogram of refractive index. I'll explain the details in the following slides. So for the last 1 year, many groups have used this HT machine for the investigate the various aspects of the science. And the professor, Yi-Bin Kang and Steve O gave a nice talk, and that has been published in Nature Cell Biology and Stem Cell Research. And there are many other topics, but for the sake of time, I'll just skip that. So the main topic of my talk is how we can combine this QPI and AI for better study of the cells and tissues. So we have developed a series of the AI application, AI-based application, including segmentation and classification and inference. And let me explain step by step. First, categorization is segmentation. This is like the segmenting the objects, such as the pedestrian or the vehicle or the buildings from the one-shot photograph that is being used in Google AI and other AI companies for autonomous vehicle technologies. And we are using the similar approaches for the analysis of the 3D tomogram of refractive index. So left-hand side, this is the refractive index tomogram of the cell images, cell culture. So now you can see the two cells in cancer cell line. And from those measurements, you can tell there are two cells and there are some subcellular organelles. But this is very qualitative analysis. In order to move to the systematic study, we thought it is the crucial to have the segmentations of subcellular organelles, including the cell membrane at the first step. So in order toward that directions, we first have measured the cell-to-cell interaction between immune cell and cancer cell. In particular, we have measured the formation of the immunological synapse between the chimeric antigen receptor T cell for immunotherapies and target leukemic B cell. And left-hand side, this is raw data, which means reflecting the tomogram measured by the Tomocube machine. And now you can see how they are interacting and how the immune cell is killing, induce the apoptosis, the target cancer cell. So this is nice to watch, but for the study, we need to provide quantitative information. So we have decided to develop the AI-based automatic segmentation algorithm. So we have trained the manual segmentation provided by the cell biologist expert, and from the 2D data of the reflecting the tomogram that has been trained for the convolutional neural networks. And after we train the various movies and cell types, and we have applied to the three-dimensional movies of the reflecting the tomogram. So this algorithm automatically performs the segmentation to tell which one is the CAR T cell and which one is target cell. The cells are label-free and live, and this is pseudo-coloring generated by the AI algorithm. So the target cell, cancer cell, was the pseudo-colored with purple color, and the immune cell was labeled, the numerically labeled by using this blue color. So since the beauty of this AI algorithm is very scalable, so we have applied to the time-lapse images as well as very large field of view by stitching the multiple field of view. And then that has been compounded by the comparing with the super resolution microscope technique. So it provides very robust and precise segmentation of the cell-to-cell interaction. And we thought we wanted to move further. So we have further developed this algorithm to segment the various subcellular organelles, including the membrane and nucleus membrane and nucleoli and lipid drumlet. And then the raw data refractiveness tomogram measured by Tomocube can be automatically segmented by using this AI algorithm. Although this is under review in a journal, but we have already launched the software, so anyone can use this software. So following by my talk, Dr. Kim will explain the details of these algorithms. The second topic is classification. It's like find the cat or dog in a photograph. So the right-hand side, you're watching the movies of the bacterial growth and divides and finally forming the bacterial film. This is again the raw data of refractive index tomogram. And as you can tell, there are a bunch of information because the different bacteria has different compositions of the protein structures and shape. And then we saw it was our hypothesis that different bacterial species can be declassified by training, by measuring this refractiveness tomogram. So this is our plan for the future application. So after taking the blood from the patient having the infectious disease such as sepsis or the other UTG, and then after taking the samples or the blood or the sputum or blood, we can measure individual bacteria. It only takes a few seconds. So 1 minute is considering the sample preparation because it takes only 0.3 seconds to measure tomogram and a few seconds to perform the reconstruction and a few seconds for the AI declassification. So the reason why we are moving towards this direction is that the conventional diagnostic process is very time consuming. And as I said, after taking the blood sample from a patient, in conventional method, you have to follow a few days of blood culture, including the primary blood culture and followed by the subculture. It usually takes 1 to 3 days. And then the current practice is to use the mass spectrometer, such as Malditov technique, in order to provide bacterial specification information. But this is not the end of the story. Even after you identify the bacterial species, you have to do culture the bacteria in the presence of various potential antibiotics called the antibiotic susceptibility test. It also takes 1 or 3 days because this is just following the culturing again. But it is our hope that since it only takes a few seconds to measure and reconstruct tomogram, and if we have trained the bacterial refractive index tomogram as of for various types of bacterial species frequently found in sepsis disease, it might be able to provide bacterial specification and identification in few seconds or minutes. And then by culturing, putting this bacteria in the presence of antibiotic condition, then we might be able to do the automatic antibiotic susceptibility test in the same time by measuring these refractive index tomogram upon few hours. So we have used the Tomocube machine. For the sake of time, I'm not going into details of the optical parts, but by scanning the illumination angles, we have measured multiple 2D hologram images of the individual cell. And by applying the reconstruction algorithm, we can reconstruct three-dimensional distribution of refractive index of individual bacterium as a function of XYZ. So we have measured at least 500 individual bacteria for species, and we have measured 19 different bacterial species frequently found in the sepsis patient in Korea. But it covers most of the sepsis patient, more than like the 96% of the sepsis cases in Korea. So we have trained the shape of this bacterium by using the convolutional neural networks. And after training, we have tested the classification algorithms. So by measuring a single bacterium, it takes few seconds to provide the answer about bacterial identification. And if we only take the one samples, and if we train the algorithm to provide one answer, the accuracy was not very high, about 92.5%. But if we adjust this algorithm to provide top 3 options, then accuracy goes up to 97.1%, but this is still not good as to be implemented in clinical settings. So we have slightly improved this algorithm such that it can take like a couple of bacterium found in one patient, not just single bacterium. And we found that by measuring more than 5 individual bacteria, the accuracy increased greater than 99.9%, which is the comparable to existing classification method based on mass spectroscopy technique. So now we are working on several hospitals to find the feasibility of this method in the clinical settings with the clinical samples. The last items that I like to explain today is inference. So the idea of inference is like follows. So if you just provide this photograph of zebras as input data, the AI algorithm automatically convert into the horse images, which is called the GAN networks, first demonstrated by the University of Montreal. And it is now widely used by the forms of the face app. I guess many of you have already used this face app to automatically convert like the face of the photograph of the male to females and young people to old people simultaneously. And we are interested in this technique because we thought it might be used to overcome the limitations of QPI. Because even though the QPI has several advantages of label-free and quantitative 3D high-resolution imaging capability, it has fundamental limitation, that is limited molecular specificity. What I mean by that, even though this QPI can provide high resolution, the refractive index distribution tomogram images from which you can tell like the overall cellular structures, but if you probe one point inside of the cell, like such as it may lead, the refractive index value may lead like 1.37, something like that. But if you ask what kind of molecules or proteins are contributing that specific values, I have no answer, no clues. Because the different species of the proteins or molecules, they exhibit the values of the refractive index for the similar concentration. That is good for the quantification, but that is bad for the molecular specificity, the imaging. That's why many existing researchers, they are combining both the QPI and fluorescence microscope and other molecular specific imaging modality in order to study both molecular information and the structural information, such as PET-CT in medical imaging. But if we can have both structural information and molecular information without staining or without introducing the effect fluorescent proteins, I think it might be good for research and for various applications. So we have employed the concept of GAN to QPI. So using our commercialized machine, we can measure both refractive index tomogram and the multi-channel 3D fluorescent imaging. So now we have access to the different modality for the same cell at same time. So we have trained this algorithm by measuring a large number of the cell data and different cell types and different conditions. And we have trained this algorithm by applying the paired data set. And once the data set was the complete, we have applied this concept to the label-free refractive index tomogram of cell. And by clicking a button, it can generate the 3-dimensional fluorescence images, but cells are still unlabeled and live and healthy. No perturbation was applied. The cells are very happy condition. And we have measured this refractive index tomogram as an input data, and this AI architecture can automatically generate actin, mitochondria, lipid drumlet, plasma membrane, nucleoli, and the nucleolus information without introducing any exogenous labeling agent. And it can automatically overcome the limitations of fluorescent imaging because there's no issue of photo bleaching and no issue of phototoxicity because we didn't introduce any exogenous labeling agent. And this is the data. Interesting thing was that we only trained this algorithm of the data set from NIH 3T3 cell. But when we apply this data set, this algorithm to other cell lines, such as CO7 and HEX cell, HeLa, MD8, and anthracite, they all provide very high accuracies and good results, even though AI haven't seen this cell type. So it implies that by training one type of cell, it provides some kind of generality for the adherent type of cell. So the question may arise, how accurate of this method compared to the conventional, the gold standard methods such as the fluorescence microscope or the immunofluorescence? So as you can see in the left-hand side, this is the process that we have done, performed to validate the accuracies. So we have measured label-free cell, live and happy. So we have retrieved the reflecting index tomogram, and we have applied the AI algorithm to infer the fluorescence information that is shown in the second column. And then in order to generate the gold standard or the ground truth data, we have followed the conventional preparation step. So we have applied the immunofluorescence. So the cells that is exactly the same cell, but you can see the cell morphology has been slightly changed due to this staining the process. Then we have measured the reflecting index tomogram, from which we apply the AI again to generate the artificial fluorescent data shown in the first column. And then for comparison purpose, since the cell has been physically labeled, we have measured the fluorescent signals by using the fluorescent microscope mode attached to the same microscope. And we have compared this AI-generated fluorescence and the physically measured the fluorescent images in three-dimensional space. They can be quantified by using the concept parameters called the structural similarity index. If you have the perfect matching in 3D space, it provides a value of 1. If there's no correlation, it provides value of 0. And for the case of NIH 3T3 cell, where we have trained this data set, accuracy is greater than 97%. And for other cell lines, where which AI algorithm haven't seen their data set before, the accuracy is still greater than 80 to 90%, which is very, very interesting. So this is not the first trial. Like a couple of years ago, the Google AI team has used the bright field microscope for the similar approaches, and they have trained the bright field data stack and they have inferred the fluorescence, but at the time the accuracy was lower than 60%. And a few months later, the Allen Institute has used the DIC microscope, differential interference contrast microscope, for the same purpose, and the accuracy is higher, but it's still lower than 70%. And when you have used the reflecting index tomogram, we have achieved accuracy greater than 97%. I think this is because reflective index information contain the bright field information as well as the DIC. They are subset to the reflective index tomogram, because in physics, the reflective index distribution describes the light matter interaction in terms of the diffraction and the scattering and everything. So you have full information. You can numerically emulate the bright field, phase contrast, and DIC, etc. So there are several advantages. For the sake of time, let me just show the data. So we can do scalable inference, and we can do long-term 3D molecular imaging. And as I said, there is no photo bleaching and phototoxicity. And if you physically measure lipid drumlet, after some time, it goes away. But AI inferred the fluorescent data just prevail over time. And then this AI algorithm is not just looking at the shape or reflective index. They look at the three-dimensional structures as a whole data set. So when we retrieve the lipid drumlet, it shows the distinct separation between lipid drumlet and endosome because even though they share the similar structural properties. And this is what we have measured using Tomocube machine over a few days of the cell culture. And right-hand side, this is all the AI-generated information. So now we can see the molecular information as well as structural information at the same time without introducing any specific exogenous labeling agent. And we can do high spatial temporal resolution study. So we have applied several dynamical, individual cell studies, and we have applied the rapamycin for the induced specific alterations. And another question may arise, can this method be applicable to other institutes where they use a different machine? So in order to directly answer to that question, after internal validation in KAIST and Tomocube, we have sent this algorithm to one of our users who has purchased the Tomocube machine a couple of years ago in other institute in other province. And they have applied this algorithm to their previously measured data set, and it has shown the good result without any modification. So which means, as you may know, there are the various AI algorithms, but you have to train again if you have different setup and different institute and different the department. But since we are using highly reproducible and highly the quantitative, the physical parameters of the reflective index, there are some general, good for the generalization. So we believe it can be used for various cell profiling the study because now we can combine the molecular information as well as structural information at the individual subcellular levels. And then it is our hope that it can be combined with the information such as genetic information and proteome information like the RNA-seq or the other special transcriptome and etc. Those are the active ongoing projects. They include the microbe and other research groups. So, so far I have briefly introduced the concept of the 3D QPI system and how AI can be helpful for analyzing the biological imagings. But there are still a lot to be improved. Like for example, now we trying to expand the applicability of this method from the individual cell study to the tissue, particularly the volumetric histopathologies. If you look at the conventional, the microscopic imaging in histological or the pathological lab, they use very thin slide of the tissue section. This is because they have to introduce H&E staining. If you have the thick tissues, it takes long time to fully label the thick tissues. That's why they just have very thin or 4 or 5 micrometers thin slide tissues. And then this is even though they have made thick tissues, the conventional bright field microscope is not good for the monitoring or imaging 3-dimensional tissue section or the organoid or the stem cell structures. But since the 3D QPI can automatically intrinsically generate 3D tomogram, we have first demonstrate the feasibility for the volumetric histopathologies, and we have measured 3D tomograms of the very thick tissues, thicker than 100 micrometer or sometimes 150 micrometer without any clearing the process. We can observe the individual cell at different cell tissue sections. And we also try to simplify and improve this optical technique because of conventional setup, or even including the Tomocube setup, is based on the laser interferometry, which means the laser beam is split by the beam splitter into 2 arms. And for controlling the illumination angles, we have used DMD, which is good for scanning for high speed with high stability compared to galvanometric meter, but still is suffer from the speckle noise issues and vibration issues, and very hard to align and to fabricate and etc. And sometimes it is not easy to maintain for a long time. That's why recently we came up with one idea in to remove the reference beam. So we were able to generate 2D and 3D QPI with like the single beam path without having any reference beam. And recently we are moving to the non-coherent light source such as LED and etc. And let me give you one latest images that we have obtained. This is still unpublished, but we have used the LED light source to generate 3D reflecting tomogram. And then this is the MV411 cell line, which is the acute myeloid leukemia. So this is individual cell. And as you can see here, this is scale bar. So resolution is around the 100 to 100 nanometer, but as you can see, the speculoid has been significantly reduced and imaging quality is significantly improved. And now you can see like the various subcellular structures, like the Golgi structures and other vesicles, and you can clearly see the nucleus membrane, etc. This is live cell, but some quality is better than the scanning electron microscope, even though the theoretical resolution is better than the optical resolution, but due to the pre-processing procedures, the cells becomes the shrunken and imaging quality is not so good sometimes, but now we can watch like this individual, the small, the object, the organelles in the live state. So I think it can open up the various interesting research applications. With that, I'd like to thank for your attention, and I'll be happy to answer to your questions.